A basic task when analyzing a single-cell dataset on TOC2 data is to explore the gene expression level. On BBrowserX, we provide a number of tools to help you visualize the gene expression of any gene or a set of genes. Now, let's learn how to use these features on BBrowserX. The simplest way to explore gene expression is to see gene expression level on the TSNE plot. By default, the scatter plot is colored by the groups of the selected metadata. When you enter a gene into the gene query box, how the gene expression across all cells is shown. The default unit is the log normalized unit, but you can change it to raw values, which is the transcript counts of the gene. Or smoothed binary unit, which tells us whether a cell expresses the gene. The gene expression of neighboring cells are taken into account to determine a cutoff value that minimizes the clustering information loss. When you query for two genes, which are considered a gene panel or a gene set, you can find out which cells express either gene or both genes with the tricolor mode. You can switch to the count mode, which shows the number of genes in the gene panel that are expressed in each cell. Or the binary mode, which tells us whether a cell expresses all genes in the panel. When 3 or more genes are queried, the count mode and the binary mode are available. You can also switch back to the metadata mode. To save a gene list, click the Save button at the bottom of the search box. All of your gene sets can be easily pulled out by choosing My Gene Set instead of typing. With a gene set, a cell is a very useful tool to see how the gene set is enriched in the datasets. You need to set a name for your analysis, then enter a gene list into the box, or choose a saved gene set. The AU cell score for each cell is displayed on the scatter plot and saved as a new metadata field. With the distribution plot, you can select cells with high enrichment scores. Next, let's produce different types of visualizations with BBrowserX. To create a chart, click on one of the 6 icons. The first type of visualization is violin plots, which provide box plots and kernel density estimations. Violin is one of the types of plots that support numerical metadata, like AU cell score, which helps you to easily compare between different groups. For the types of plots that support the split by function, you can select a second metadata field for better comparison. The second type of visualization is bar charts, with the error bars showing the 95% confidence intervals. The third type of visualization is bubble heat maps. The sizes of the dots represent the percentages of cells that express the gene or the coverage. And the color represents the level of expression of the gene in the datasets. The darker the color, the higher level of gene expression. When the scale mode is absolute, the sizes of the circles are scaled from 1% to 100%. Change the scale mode to relative to scale the sizes of the circles from the smallest value to the largest value. This mode amplifies the difference between two values. The fourth type of visualization is circos plots, which display the expression level of a gene in a group by a flow. The fifth type of visualization is intersection plots, which require at least 2 genes. In an intersection plot, or upset plot, the matrix at the bottom shows different combinations of the input genes, and the bars represent the number of cells that express the respective combinations. The last type of visualization is co-expression heat maps. Co-expression heat maps show the level of co-expression between pairs of genes within the dataset based on the Jarkart score. When you query a list of genes, the co-expression matrix will show the co-expression level of all the combinations of 2 genes from the list. When you query for one gene, the co-expression matrix will show up to 50 genes with the highest co-expression levels in the atlas with the gene you query. Click on a dot to display the Venn diagram and the correlation plot of a pair of genes. Each chart can be exported to Vinci for editing. Besides this section, BBrowserX allows you to visualize gene expression directly on a composition chart and create advanced interactive heat maps. We hope you find this video helpful. If you have any questions, contact us anytime via support@BioTuring.com.